Bismillahirrahmanirrahim. In the name of Allah, the most beneficent and the most merciful. Dear students, Assalamu alaikum wa rahmatullahi wa barakatuh. Dear students, I welcome you in Pakistan International School TAI's virtual learning classes for the session 2020-21. And you know that this is class for biology for grade 9 junior, and we are studying chapter number 5, cell cycle. And today's topic is very important, very important indeed, that is meiosis. So let's get started. Dear students, in the previous lectures, we have finished mitotic division or mitosis, in which one cell is divided into two new cells. So the old cells is said to be mother cell and two new cells are said to be daughter cells. And this is according to the third postulate of cell theory, omnis cellulae e cellulae, like new cells arise from the pre-existing cells. So this mitosis process is very important throughout the life, especially in the childhood stage, when whole body cells, or those are called somatic cells, they undergo mitotic process by which the cells grow, your bones grow, your parts of the body grow in size. Today we will talk about another category of cell division that is called meiosis, and meiosis is complete. It is also cell division, but it is completely different from mitosis. We will see its differentiation also in the next lecture, inshallah. But right now, we will see how meiosis is done, where it is done, why it is important. Meiosis, you can see here in the diagram that meiosis, when this is happened, this undergoes through the same cell division as the mitosis is done by, but it is given different result. Like it undergo first its interphase, then all the processes are same as mitosis, but in that process is there is also some differences. And in the last, you can see here there are four cells. So actually, meiosis is the process. Meiosis is the process of cell division in which diploid eukaryotic cell divides to generate four haploid daughter cells. Now, there are some terms to remember because I have discussed this in the previous lectures also. Number one, what is diploid cell, eukaryotic cell, and haploid cell? And what are daughter cells? So, diploid means, di means two, ploid means number of chromosomes. So it means 2n. 2n is the number of chromosomes and 2 comes from the double number of chromosomes. So eukaryotic cells, you know, in that the nucleus is completely present in its complete form, like it is having the cell membrane, nuclear membrane around the chromosomes. So like human being cell, like plant cells, so eukaryotic cells, one diploid eukaryotic cell is divided into four haploid daughter cells. Daughter cells, the cells which are newly formed, they are called daughter cells. As I mentioned before, the, the old cell is called mother cell and the new cells are called daughter cells. So haploid, hap comes from the HA comes from the word half, and ploid means the number of cells. So as many number of chromosomes, n comes, or ploid comes from the number of chromosomes. So as many number of chromosomes are present in one cell, when it is divided into four, definitely it will go into the half number of chromosomes. For example, this cell, this is a parent cell or mother cell, then it is going through all the processes of cell division, and after that it results into four number of cells. From one cell, four daughter cells are formed, and in there you can see the structures of the chromosomes. So, like here, one, two, three, four cells, chromosomes are present in one cell. So here in each cell, two chromosomes are present. So it means if human beings are having 46 chromosomes, so when it will divide into four cells, so it will be 23, 23, 23, and 23. How it will happen? All this thing, like 46 chromosome have to be divided into 23 and 23. So these are like in first stage, it will divide it into two cells in 23 numbers of chromosomes. And later, when these cells divide, they form four cells which are having all 23, 23, 23, and 23 number of chromosomes. It means 46 is diploid number of chromosome and 23 is haploid number of chromosomes. So in the next classes and in this chapter, in the next topics, we'll also use this term diploid and haploid. So you must remember these terms. What are these? So diploid means the cells which are having the chromosomes in pairs. Definitely when they are having, like for example, this is one type of chromosome. So its pair is this one, like exactly, I can't make exact, but they both are having exact like shape and structure, so they form the pair. So here you can see they form the pair, but when they are in the last four chromosome number, then they will be alone, no pairs are there. So that's why they are called half number of chromosomes. So how all this process is going on, we will discuss it in the next slides. Here is, you can see two n numbers like diploid number, mother cell or parent cell is having, then this one, and in the last when we'll get like it is divided into two, then it will this will divide into two and this will divide into two further, so that they are called haploid number of chromosomes. So now we will see that meiosis was discovered and described for the first time in 1876 by the German biologist Oscar Hartwig. He discovered this term meiosis. So what does this term mean? We will discuss it. Like this term means to get shortened, reduction. Actually, this is a Greek word. It means meon, and its meaning to make smaller, since it results in a reduction in the chromosome number. Means chromosome number reduced, so the number of chromosomes gets smaller. That's why this process is said to be meiosis. Okay, let's talk about how this process is going to be done. We will see for that phases of meiosis, like step by step, we will discuss that. So first of all, we will see what type of phases of meiosis are there. So for that, we know, so meiosis or mitosis, for the cell division, the cells is going to prepare itself for the division process. For that, they have to take a rest for some time, and then they go through a phase that is called interphase. So in interphase, inter means in between, phase means the stage between two mitotic division or meiotic division, the cell goes in between a preparatory stage. So preparatory steps for meiosis are identical to interphase of mitosis. So here we are having the same stages, G1 S phase and G2 phase. So you know that what is going to be happen in G1 phase, S phase, and G2 phase. There is the enzymes and organelles preparation and formation, and the cell size increased in G1 phase, and and that is required for the preparation of S phase. And S phase is synthesis phase, G1 is gap 1 phase, G2 is gap 2 phase. So in synthesis phase, the chromosomes are duplicated and they are getting preparation for the next division stage. Because when they will be duplicated, then they will have the same number of chromosomes in the next division stage. Then they are having the cells are having an interphase stage, G2 phase, in which they are preparing enzymes for this stage. This is called meiotic stage. So the cell has gone after interphase two stages. These are called meiosis 1 and meiosis 2. For meiosis 1, it is divided into two cells, and then in meiosis 2, both of the cells divide again into two cells, so they will get four cells. So what happens in meiosis 1? Actually, the homologous chromosomes in a diploid cell separate, so two haploid daughter cells are produced. First of all, the chromosomes are condensed. They are, like we'll see in prophase 1. So in meiosis, the homologous, homo means same. So the same chromosomes make pairs, and that pair is called homologous chromosome. After division, the diploid cell, like in which the chromosome numbers are doubled, then we'll get two half number of chromosomes which are present in two cells. So it is the step of meiosis in that generates genetic variations also. Genetic comes from the word genes. Like genes are the sequence, or we can say a smaller part of DNA, which is present in the chromosome, and it get variated, like it gets change here. So this is very important term because it will help you in the next classes also. So you must remember what is genetic variations, because uh, how you are different from your parents, this is actually the genetic variations. Some are having like eyes with resembles with the mother, but hair with the father, and some like totally different. How this all happens, like how the next offspring is different from the parents, that is called genetic variation, like change in the genes. Genes are not changed, but how these variations is occurred, we will talk about in the next slide, inshallah. So in meiosis 1, it occurs in two main steps. First of all, like mitosis, it's karyokinesis, and then cytokinesis, in which the nucleus is divided and cytokinesis, cytoplasm is divided. So karyokinesis or meiosis is subdivided into again prophase, metaphase, anaphase, and telophase. But because this is meiosis 1, the first division stage of meiosis, so that's why we call stages of that prophase 1, metaphase 1, anaphase 1, and telophase 1. So here you can see how the cells are divided into four cells. First of all, they divided into two, then these two cells like this, these two, and then these two cells undergo again one more division, and they are called four cells now. Now we'll talk about first stage of that, prophase 1. So prophase 1 is the longest phase in meiosis, and during this stage, chromatin condenses into chromosomes, because chromatin is duplicated and then it is condensed into chromosomes. So here the same chromosomes, the form pair, they're called homologous chromosomes, and they line up with each other to form pairs, and those pairs are called synapses. By the process that is called synapses, how they form pair you can see like this. This is in mitosis. We have seen this type of structure, this one and this one. So here is the centromere, both of the sides are kinetochore. Okay, so this is actually one chromatid and two chromatid. So after synapses, when the same chromosomes they get duplicated, like these are homologous chromosomes, then they form pair with one another. So this process is called, when they are pairing with one another, that is called synapses. Remember this thing. When the homologous chromosomes, this one purple and the blue one, other homologous chromosome, they line up with each other and they form pairs. So that is called synapses. These are same, so they are called sister chromatids, but the blue are same, both blue chromatids, so they are called but the purple and blue one, they are not same, so they are called non-sister chromatids. So you can see here the centromere, and these are kinetochore proteins. So both of the sides, both of the sets of the homologous chromosomes, they are having kinetochore protein. So each pair of homologous chromosome is called bivalent. Each pair of homologous chromosome, this one purple and this one, they are called bivalent. Bivalent. Okay, like this one pair, and this one pair, both they are two. Now they are called bivalent, and each bivalent has four chromatids. Now you can see this number one, number two, number three, and number four, this blue one, two blue chromatids and two purple chromatids. So they are called four chromatids. So now four chromatids, due to four, they are called tetrad. Tetra, with tetra say four. So four chromatids. So here in the nucleus, you can see these all chromosomes line up together and they do synapses, and now there are tetrad. Okay, so after that, the two non-sister chromatids of homologous chromosomes of each other at certain point along their length. Now what they do, they join each other, non-sister chromatids. Here you can see these blue one and purple, they are non-sister chromatids. Now they are going to join here at this point, and this point of attachment is called chiasmata. Chiasmata. This point of attachment, this one where they are joining, that is called chiasmata. In the next stage, the non-sister chromatids of homologous chromosomes exchange their segments. This phenomena is called crossing over. This is very important step in meiosis 1 prophase 1, because due to this process genetic variations occur. It means here the genes which are present here at this place or at this place, they are different because they are non-sister chromatids. They here the genes are different and here the genes are different. So when they cross over one another, like they form cross, and they like now blue part just become the part of purple, and purple part become the part of the blue chromatid. So when they get exchange their a part of the chromosome, that process is called crossing over. And crossing over is very important for genetic variations, which we have discussed in the previous slides. Genetic variations, the genes come from one non-sister chromatid to the other non-sister chromatid. So when they will get separate, so different gene, which were the part of this purple one, it is connected now linked with the blue one. So they will go in one cell and they will go in one cell. So here the genes are different now. So here you can see very good example of genetic variations also. Here you can see the red one and blue one. You can see here. Look, this one. It can be on one place or it can be on two or more than one place. Look here and here at the back. You can see carefully with this diagram. So all four chromatids, non-sister chromatids, are exchanging the genes. So as many genes are exchanged, so as many variations in the next offspring will be produced. So the exchange of segments result in the recombination of genetic information. Recombination comes from the word first combine and then recombine, like genetic information is recombined with one another. So when they are recombined, or genetic information is done after crossing over, each pair of homologous chromosomes uh, now remain as a bivalent. Still, they are present in their form. Here, the look you can see the materials has been exchanged. There a genetic recombination is done. Recombination means they have formed their genetic information again. So they have exchanged their genetic information, but they remain still at this bivalent position, or that is called tetrad position. So chromosomes condense further. You can see this is the nuclear envelope. Now nuclear envelope disintegrates and you can see centrioles which are duplicated before in interphase. Now it is going to be migrate to on both of the sides of the nucleus to form spindle fibers. So the kinetochore spindle fibers attach with the kinetochore of chromosomes. We know that spindle fibers are having kinetochore protein also, so they are connected. You can see here, or you can see here with the kinetochore of the chromosome. While the non-kinetochore spindle fibers from both sides interact with one another, kinetochore proteins, kinetochore spindle fibers attach with kinetochore of chromosomes, but if they are not kinetochore, so they are not connected with one another and not with the chromosomes, but they are connected with one another in the with the spindle fibers from both of the sides. Two kinetochore spindle fibers from the opposite pole attach with the pair of chromosome. In meiosis, if you see one kinetochore protein and the other kinetochore spindle fiber, two are attached with the whole pair of the chromosomes, not separately. This one, this one, this one. They're not four, but just two kinetochore spindle fibers are attached, whole of the pair. But in mitosis, we have seen that two kinetochore spindle fibers attached with one chromosome. This is chromosome with one chromosome, and on both of the sides of that chromosome, the kinetochore spindle fiber is attached. So this is also the difference between mitosis and meiosis. So next stage, this was very important and longer stage, that is called prophase 1, in which the genetic recombination has occurred. So that process is called crossing over. So that is done in prophase 1. All other stages are similar to mitosis. Like that, the pair of homologous chromosomes align, like the pair of chromosomes align along equatorial plane, forming the metaphase plate. So here you can see metaphase plate, they're attached here. And after that, when they attached over there, the next stage will be started. So in 19, very important, there's just information that is given in side box in your book. In 1911, because we have seen about the genetic variation, so the American geneticist Thomas Hunt Morgan observed the phenomenon of crossing over in fruit fly Drosophila melanogaster first time. That how does the crossing over in the fruit fly. Fruit fly, this is a fruit fly which lays eggs in the fruit, especially like we can say the oranges, guava, and like that in the fruits. It penetrates in the fruit and then lay eggs over there. So that scientific name is Drosophila melanogaster, and he is Thomas Hunt Morgan. He first time observed the process of crossing over in this fly, which we have seen before. Okay, so after metaphase stage, the next anaphase stage will begin. And in that, we know that kinetochore spindle fibers shorten, and it results in pulling apart of chromosomes of each other. So like you can remind here or remember here anaphase. So mitosis the same is there, but somehow different because in that one chromosome has been divided, but here the pair has been divided now, and two haploid sets of are formed. Haploid sets of chromosomes are formed, which are pulled by shortening of the spindle fibers on both of the sides. Then we are having the next step. What is that? The last one of that, that is telophase. So in telophase 1, chromosomes arrive at the poles. Each pole has half number of chromosomes because yet they are in the pair. So still consist of two chromatids, like in the mitosis, mitosis in the start. When prophase, just remember prophase of mitosis, this is exactly the same now. So nuclear envelope is developed, chromatids or chromosomes gets uncoiled and they become chromatin material. And then the next stage of mitosis has been begun, but after the cytokinesis, the nucleus has been divided. So after that, the in animal cell, that cytoplasm is pinches from the center, and then um, it divided the cytoplasm into two cells by having these haploid number of chromosomes on both of the sides. But in plant cell, we know that Golgi vesicles just align in the center, and they split in the cytoplasm, and cell wall from the center, a new cell wall will arise, and then the cell is separated into two cells. In plants, meiosis is also done. Now we will see this up to cytokinesis, that is called cytokinesis 1. So up to cytokinesis 1, we have completed meiosis 1. Now we will see mitosis 2, because after meiosis 1, we get two cells. Now after meiosis 2, we'll get four cells. So um, between two divisions, cells will also take rest for interphase stage. So after meiosis, both of the haploid daughter cells will get rest. That is called interkinesis. Kinesis means breakdown, and inter means between two divisions or breakdown, that is called interphase 2. Remember one thing, interphase 2 is not the same as interphase 1, because in interphase 1, S phase is there in which chromosomes are duplicated, but in interphase 2, S chromosome is not duplicated. So remember this thing, this is interphase 2. So that's why they remain haploid in the four cells. Chromosomes numbers don't get duplicated. So then we'll talk about meiosis 2. So this is the second part of the meiosis and similar to mitosis. So that's why this meiosis 2 is exactly same mitosis. So it is subdivided into prophase 2, metaphase 2, anaphase 2, and telophase 2. So in prophase 2, the same thing is happened, takes much time as compared to prophase 1, and the same nucleoli, nuclear membrane, chromatin material condenses, but all the things are disappear. So centrioles move on the both of the sides and make spindle fibers. In metaphase 2, chromosomes attach with the kinetochore spindle fibers and align at the equatorial plate. Then in anaphase 2, the centromeres are cleaved. Centromeres are cleaved means breakdown and sister chromatids are pulled apart, and then sister chromatids are now called sister chromosomes, and they are pulled towards opposing poles. So telophase 2, uncoiling of chromosomes is done into chromatin material, nuclear envelope reforms, cleavage of cell wall and cell membrane is done, and we will get four daughter cells in which all the cells are having haploid set of chromosomes. So now we are having summary of this one. You can see the prophase, first of all, prophase 2 stage. You can see here the same as it is mitosis. So nuclear membrane disappears and centrioles moving on both of the sides. So this is homologous chromosomes. They are aligned at the equatorial plate. Now in metaphase 2, you can see the metaphase 1. This synapse is going to be aligned in the center, but in metaphase 2, there's two chromatids, that is called one chromosome is aligned over there. Now we will see anaphase 2. So you can compare that. In anaphase 1, we have four chromatids which are get split now, and they become two chromatids. And in anaphase 2, now at this stage, which is similar to mitosis, one chromatid is there. One chromatid is there. So you can see the haploid number of chromosomes now. In telophase, all the things are reappeared, cell membrane and like a nucleolus, and chromosomes just get uncoiled, and they are having half number of chromosomes, and we get four cells, 1, 2, 3, and 4. Here is the summary of meiosis. You can see here one cell, it is divided. This is prophase 1, metaphase 1, anaphase 1, and telophase 1. Here, that is not all the detail, but here we can see that this is the first division, meiosis 1, and here this is called meiosis 2 division. At the end, we'll get four haploid cells in which the chromosome number in human beings, that is 23 for each. Here we get 23, then this will divide into further 2, 23, 23, and this will, it means in the first meiotic division, we had haploid number of chromosome like 23, and in meiosis 2, that is exactly same to mitosis. So in mitosis, the number of chromosomes remains same. So before it was 23, now in all the cells we'll get 23 number of chromosomes. Remember one thing, this meiosis is done in germ-line cells, like in the gametes, where male sperms and female eggs are produced, and then they may do fertilization, and a new embryo is formed, and new offspring is going to be generated here. So dear students, that's it for today. That was sorry for the taking too much time, because that was the concept which cannot which cannot be divided into two different lectures. Wish you all the best. Inshallah, we'll continue this one in the next lecture by the new topic about significance of meiosis, and then we will have differentiation between mitosis and meiosis. Wish you all the best. See you then. Allah Hafiz.